I think we can get going because Bonnie confirmed that she sent everybody over, so they should filing in. So I just want to take a moment to welcome everyone again to SAMPS Networking. I want to thank our sponsor, BioCompare, tonight. If you haven't already had a chance, go over to the Expo section and take a look at BioCompare and what they're all about. And if you already haven't done that, I recommend you go over there. And there's also a place for more information about SAMPS if you're interested in finding out more about SAMPS. For the regulars of our events who've been joining us throughout this past year, you'll notice that we're trying something a little bit different tonight. So we have a guest speaker, and we're combining a webinar in here as well. So without further delay, I'd like to introduce Dale Yuzuki. Dale's a business development, market development, and sales professional in the oncology testing and liquid biopsy markets. And he's also a board member at SAMPS. He's the author of the book COVID-19 From Chaos to Cure. And Dale's going to present timely and relevant information tonight about the status of the COVID-19 vaccine development, the design of the clinical trials, as well as his own personal experience with the phase 2 Novavax vaccine. So welcome, Dale.
Well, thank you, Mark, for the opportunity to share this afternoon. I know it's a little different format because it's only a one-way communication. Certainly feel free to use the chat box. Mark will monitor that. And then we can address questions later in the networking session where we'll set up a Q&A session. A little bit about myself, 6 years, maybe I'll back up even further. 2 years as a high school student, another 2 years in graduate school with cell biology, master's degree, 6.5 years HIV and cancer research, a couple years spent abroad, and the last 20 years involved in life science, everything from technical support at QIAGEN through product management at Illumina, through sales for 6 years, both Illumina and Raindance, through more sales with Life Technologies, Thermo Fisher, Ion Torrent, and then regional market development and social media development at Thermo Fisher, and then a clinical turn at SaraCare, and then a couple of startups the last 3 or 4 years, including Sysmex Diagnostics and Singlera, which just received $150 million in funding. It was about June or July, I had been learning so much about COVID-19 and thinking about how much poor reporting was out there in terms of diagnostic testing, in terms of the state of therapeutics, as well as vaccine development. I thought I'd write a book from an insider perspective of how all these three pieces work, and then how they're all dependent upon biotechnology and the remarkable advances in biotechnology in the last 50 to 60 years. So today, I thought I would take a little different tack, and that is one, to talk a little bit about how difficult COVID-19 diagnostics are. Devilishly difficult. In the book, I talk about the intricacies of the immune system and how that immune response, as well as the preconditioning of native immunity, makes some people less susceptible to COVID-19 disease once they get infected. But there's some real diagnostic challenges that aren't going away. And also to give some updates on new diagnostics. And then we'll talk a little bit about therapeutic advancements. Certainly in the last couple months, there have been some exciting approvals for therapeutics, as well as some things that are, how do I say, very interesting developments from some of the controversies that plagued us over the summer. Then we'll talk about vaccine progress, including my own experience. And then, like I mentioned, questions and answers will be handled separately. So this is a figure from figure 5 in the book, that's from the reference below, about the kind of detectability of the virus itself. So you see, a full 2 weeks before your symptoms start, you can be exposed, right, to somebody who's infected. About 5 to 7 days before you actually get symptoms, you will have live culturable virus in your upper respiratory tract. Okay, sometimes it travels down quickly, but the majority time it's in your respiratory tract, and of course it's then transmitted through, as we now know, through aerosols as well as droplets. This presence of live virus, as you can see, can occur, like I mentioned, 5 to 7 days before you have symptoms, and once you have symptoms beginning, this is day 0, it is this part right here, this asymptomatic spread. People who don't know that they're infected, they go on infecting others. This is what makes it so difficult, in addition to whether or not people even have symptoms, because there are cases, right, where you have asymptomatic individuals with live virus, and still there's a lot that is currently not known in terms of how infectious are the asymptomatic, as well as the factors that go into someone that is able to cause a super spreading event. Now, one thing that came up, that this was just recently, this is work done at the University of Maryland that looked at infectability of individuals using an influenza model. So this is just the flu transmission. And they found out that, frankly, it was about 5 out of 40, so about 25%, if my math is correct. Yeah. About 25% of individuals, for whatever reason, are really good at aerosolizing. That is, through their construction of their vocal cords and their larynx and their basic physiology, their mouth equipment, so to speak, they aerosol a lot. And it's also interesting to note that it is a bit of a Pareto Principle. This was about 2 months ago, a study out of Princeton looking at contact tracing of over 170,000 cases in India, demonstrated that 60% of the infections was caused by only 16% of the individuals. So you have this mismatch between who's infecting everybody, right, in terms of a relatively small percentage of people infecting a lot of people. And one article that you should look up is the K factor. It's an Atlantic article looking at the transmissibility. It's an overlooked factor in this epidemic, in this pandemic. We're all familiar with R0 and the transmissibility ratio, basically looking at the rate of exponential spread. And this is referring to if it's greater than 1, right, you've got an exponential growth where, if it's 2, it's 1 person infecting 2 people, those 2 people infecting 2 others. And if it's less than 2, then you get less than exponential spread and it will cycle downward. If it's greater than 2, it's exponentially growing upward. Well, that is assuming, making a big assumption that every individual who gets infected in turn infects 2 others. Well, the K transmissibility model is a little bit different because now you're looking at, frankly, super spreader events from a minority population. You're looking at the 20-25%, or in the case of that India contact tracing, only 60% of individuals, accounting for 60% of all the cases. And so this is a matter of the way this virus transmits, where some people are highly susceptible to infecting many others at once. Anyway, looking at this figure again, okay, you've got a couple of difficulties here, which is people not having symptoms and then they infecting others, and then is compounded once again with the sensitivity and specificity of molecular testing, okay? Of course, you can see here the presence of viral RNA that lingers on for weeks, even though the person may not be able to actually infect others, right? And then, of course, there's the serological response in terms of antibody testing goes. So for the next slide is an interesting one. And this is looking at false negative rates using RT-PCR. And one of the challenges in this meta-study, okay, was looking at RT-PCR performance by time of symptom onset. So they tried really hard to get samples before people had symptoms. and then collate this data together to look at the chances of having a false negative. That is, your SARS coronavirus 2 positive, but you're PCR negative. All right, think about that. You have the virus, you are infecting others with the virus, but the chances of it being PCR negative are 100% way early before symptoms appear. As you can see, the day that symptoms appear, you still have a 38% chance of being PCR negative but coronavirus positive, and it goes down there, thankfully, right, to where it's in the 20% range. But still, you're looking at a 20% false negative rate by RT-PCR, but you're coronavirus positive in terms of culturing virus. Okay, so we're looking at within the 7-day window, right, of having symptoms. So it's in this area in bold. Thankfully, it's relatively low, but still, you've got a PCR negative rate just because of the nature of the virus, as well as the sensitivity. You can say, well, you've got molecular sensitivity on the order of maybe 35 or 38 PCR cycles, and they validated these clinical tests down to a handful of molecules per mil of viral transport fluid. And of course, we won't go into all the variability in terms of metrics. Are you talking about molecules per mil of fluid? Is that 3 mils of viral transport media, et cetera, et cetera? So a lot of details to wade through. Even talking about the Bayesian model, I was looking at some of the comments of this article. It's pretty interesting in terms of mathematical model. But nonetheless, on the day of symptom onset, this particular paper measured a false negative rate of 38%. And there's a lot that gets baked into that 38% false negative number, but suffice it to say, the individual's response to the virus is varying widely, and the sensitivity of the tests, its molecular sensitivity is good. Like I mentioned, the Quest molecular test is down to like 6 molecules per mil of VTM. But nonetheless, right, there are difficulties in detection even using gold standard PCR. And the conclusions of the paper was you can't depend on just a molecular test for diagnosis. You need to have the other markers of infection. Okay, going on a little bit further in terms of the testing field, one of the interesting at-home tests that was recently approved, this was approved in November, this is for a device and consumables for a molecular test at home. Now you think about what it takes to do RT-PCR at home, and what this company did out of Emeryville was develop an isothermal PCR method called LAMP. And LAMP is a difficult assay to design for. It has some specificity kinds of challenges, but nonetheless approved, right, for home sampling. And the only caveat is that you need a physician's prescription in order to get both the instrument and the reagents for it. Pretty interesting concept, battery operated, isothermal, right? It's just a heater, and you basically, I cribbed these notes from the instructions for use. You stir the sample vial 15 times, you discard the swab, you snap the cap in, and you plug it in the device, and this will automatically read colorimetrically a positive or negative or an invalid result. Pretty remarkable in that you swab, you place in the unit, and you read the answer in 30 minutes. Now this is going to help, certainly, in terms of getting wider based testing. I know thought leaders like Michael Mina, right, have been out there being quite verbal, Scott Gottlieb as well, quite verbal in saying we need to have, less sensitive tests, but really widespread. And I cover this in the book, but the short answer is, our system here in the U.S. is not set up for surveillance, right? We do surveillance for a newborn screening, for CF testing. That's a certain types of surveillance. We have surveillance for TB and other diseases for the CDC. but to have, all Americans undergo a diagnostic test and basically to screen out those super spreaders is the goal, right? Is to screen out these individuals here through fast and cheap testing on a widespread scale. We kind of missed the boat, meaning we had the opportunity to do something like that if the tests were available and if the will and the direction were given to do that kind of thing. But there are other kinds of harms, right? When you, in terms of the cost of false positives, the cost of false negatives, I think that's going to be a debate for years to come, especially as we're thinking about, right, preparing for the next one. And it's just a matter of time, right? When you look at the history of pandemics, especially with coronavirus, first in 2002, and then again with MERS in 2010 and 11, and then Ebola in between there. And so we've had these recurring issues where diagnostics and particular point of care testing or at-home testing is really needed. Okay. All right, a quick update on therapeutics. A lot of progress has been made. Okay, there have been three monoclonal antibody EUAs, all right? First from Regeneron, there's a cocktail of two. Another one from Eli Lilly, another cocktail of two. These were both similar in that they were for mild and moderate COVID-19 disease at high risk to progress to severe. So these are people with a known risk factor. I think that covers quite a few individuals in the U.S., right? I think it's maybe on the order of maybe 40 or 50% have at least one risk factor. And then you're looking at either for the Regeneron, a significant reduction in viral levels in 3 days. And Eli Lilly, they were measuring return to hospital, meaning they had mild or moderate COVID. They gave the placebo and compared to the treated group, and 10% versus a 3% hospitalization rate. So they're able to cut down the hospitalization rate by a factor of 3, which is significant, right? And you think about it, right? The ability to prevent people from going from mild and moderate disease to hospitalized means you're saving lives, right? And they're also looking at this as a temporary vaccine, that is to provide prophylactic protection against people at high risk, such as first-line responders. Another therapeutic is an antibody as well. This is a drug, but it's a JAK inhibitor, which is kind of an interesting repurpose of a cancer drug. It interferes with the inflammatory pathway, and they administered it with remdesivir. And what it did was it reduced the time in the to recovery from 8 days to 7 days. So it's not great, but it was still significant, statistically significant. And they also lowered the odds of progression to death. And so that was also an improvement. Not a panacea by any means, but nonetheless, repurposing an existing drug with remdesivir is seen as a plus. One handy URL is at raps.org. This is a regulatory professional society. They put together a therapeutics tracker that's really convenient to get at a glance what is being developed and where. And this says 10 entries, but there's, I think, about 70 in their database that they're keeping track of. And then for vaccines, they have a vaccine tracker as well, and there's over 200 vaccines in there. So it's pretty remarkable when you think about the effort around vaccines. This is another figure from the book that lays out the time it takes from development to approval for a vaccine. The average is about 14 years. You can see the world record was in 1967 for the mumps vaccine. It only took 4 years. And this is why at the beginning of the pandemic, a lot of scientists were pretty skeptical in order to get a approval by the end of the year, even mid-2021 was considered iffy, given, the world record was 4 years. But I can say, as someone who used to sell the NIH and have been at the vaccine research centers at Building 10 or Building 11, not Building 10, it's like Building 12 on the NIH campus. They were on the phone the day after the genome sequence for SARS coronavirus 2 was published in China. And they were on the phone with Moderna and with Novavax and others as far as discussing their existing approaches and whether or not they could quickly pivot to work on producing a vaccine for the novel coronavirus. And it's clear, right, with the Pfizer that you probably are familiar with, these are some of the details. One of the remarkable things was this, right, the endpoint of severe COVID. If you're able to reduce the amount of people getting severe COVID, you're saving lives right there because they're not going to progress to death, right? Another remarkable thing was the number of minorities they enrolled. I mean, out of 44,000, that was a lot. I think the other, of course, the headline, right, where you've got 95% effective at preventing infection. And then the study, they only required one symptom to be considered infected, okay? And then, I'm sorry, as far as the side effects, right, they 3.8% report fatigue, 2% headaches. One of the challenges with the Pfizer vaccine, as you may be aware of, since it's an RNA vaccine, it needs to be handled on dry ice or -80, and that's going to be a challenge. Now, Pfizer developed a very compact, it's like a 16 by 24 inch by 16 inch box that can handle 5,000 doses and hold it at 10 days shipment time. And then they've given guidance as far as how long it's stable at either -20 or at 4 degrees. So that's currently being rolled out. On Moderna, what's remarkable with this one, right, was that, yeah, it was 94.7% effective, 11 out of 95 were severe COVID. But think of this, of the 11 that had severe COVID, all 11 were in the placebo group. What does that mean? 0% developed severe COVID. And so this is absolutely remarkable, as the Science News article points out. And then the study required 2 symptoms to report. The symptoms were a bit higher. Instead of 3.8%, and the Pfizer, you've got 9.7%. And this is similar to a shingles vaccine that was recently approved, in terms of about 10% of individuals reporting some sort of side effect. The transportation and storage is not as big of an issue because it's at -20 and it's stable at 30 degrees. Moderna, although the results for the phase 3 has been announced, it has not been approved yet, although it may be just any day now. Another one you may have heard of was AstraZeneca, 70% effective. The US study is ongoing. This one's different. The first 2 you probably know are mRNA vaccines. This is a chimpanzee adenovirus, and the idea and rationale behind using a non-human adenovirus is that people are very unlikely to be pre-exposed to a chimpanzee adenovirus. The thinking was, if you already had adenovirus infection, you have antibodies against the adenovirus, and there is a risk that of something that was called ADE, or antibody-dependent enhancement, where people who took the dengue vaccine, once they were exposed to dengue, the actual dengue fever virus, they got worse symptoms than if they had nothing at all. And so that was something that the scientists were very careful to take a look at, both with the mRNA vaccines as well as this one. But nothing like that has been observed. They haven't observed any severe COVID, and then they've got a better transportation mode at 4 degrees. Worldwide status, you can see here, these are the 1, 2, 3, 4, 5, 6 viruses that have been approved worldwide, with the Pfizer at the top, and then these are Russian or Chinese specific vaccines. This one is the up and coming in phase 3, so the Moderna one was mentioned. This one is the University of Oxford AstraZeneca one was mentioned. J&J, you just don't hear much about, but it's still moving its way through. And this Novavax one is in phase 3 now, and this is the one that I took. I was part of a phase 2B. I got severe side effects. I would say severe in that I had a headache, and my arm was really sore on the second dose, and I felt bad for about 2 days. I think the myalgia was probably the worst I've felt in a while. It's kind of like a bad cold where you're just, muscles are hurting and you just want to lie in bed all day. And that's what I did for maybe half a day. It's just moving around. Wasn't all that great. Wasn't debilitating, right? It was just a nuisance, and it went away after 2 or 3 days. A few endnotes. If you go to this bit.ly link, there's an interview with me about what the book's about. I've got an email newsletter to sign up, and if you provide an Amazon review, I'd highly appreciate it, because very, very few people write reviews. I'm finding out firsthand. And lastly, this is the book cover. Just published a few weeks ago, and it got some nice things for people to say in terms of digging below superficial data provided to the general public. I mean, I wrote this for the curious layperson, right? I did not write this for the expert molecular biologist, and was able to get into some of the history of DNA and things like that, as well as how basically the miracles of biotechnology were brought to bear in diagnostics, in therapeutics, and in vaccines. And this person was well-written, exceptionally detailed guide explaining history, background, and current research. And I also had to write it with the eye to, okay, well, is this going to be relevant in 2 years, right? And certainly, you can see the therapeutic paths being approved, you can see new diagnostics coming online, and you can see the vaccine as being the ultimate cure and end of this. And as you all know, right, it's now being rolled out. Probably, was it by April, I think, Dr. Fauci said that very likely everybody in the U.S. who wants a vaccine would be able to have one. And I think that I myself am pretty optimistic in that that point of herd immunity, of where the number of people who are naturally immune, in addition to people who get the vaccine, it may be sooner rather than later. We may not even need to get to 60% vaccinated, because I think there's growing evidence that a decent percentage of the population exposed to the common cold coronaviruses actually have cross-reactivity to the novel coronavirus that causes SARS-2. So, well, with that, I invite you then to go over from the stage on your left to hop over to one of the breakout sessions, and I see Mark. Going to help me with that.
Yeah, absolutely. So over on the left, you'll see sessions, and now says now because it's 5 o'clock on the East Coast, and now that those sessions have opened up. So your choices now are to go into a session with Dale, where he'll take questions and field any concerns that you have, and maybe he can help out with some of the questions you have on this. And then you also have a choice to go to just an empty room that we have off to the side if you want to just chat with people, or again, you can go to our speed networking and do the one-on-one networking. So I just want to thank Dale. I thought that was excellent, given that we only gave you 30 minutes to do this, but I'm sure there's going to be a lot of great questions in the follow-up session that's going to occur now. So thank you, Dale.
Great. Thank you, Mark.